Okay, fine. Let's look at example number 2. I will give you 3 examples from now. Example number 2, number 3, and number 4. Let's quickly look at example number 2. In example number 2, they are saying a purebred white-faced cow was crossed with a heterozygous white-faced bull. White is dominant over black. Use a Punnett Square to show the F1 generation. Use letter Q for the white color. So you are given a statement. I would like to focus on the first 2 sentences now. You are given a statement. You are supposed to draw a Punnett Square to show the F1 generation. So you cross the 2 parents to show the F1 generation. Before you do that, you must be able to come up with the genotype of the 2 parents. Here we're only told the phenotype of the parents, not the genotype. But when you do a square, a cross, you must use the genotype. So our task now is to come up with the genotype of the bull and the genotype of the cow. The first thing that we must do, let's read again. A purebred white-faced cow. Let me just stop here. A purebred white-faced cow. It's a cow that is white. And the alleles, they are in purebred. We said when we talk about the homozygous, purebred, they can also use it to show what? A homozygous. What do we mean by homozygous? We simply mean that the alleles are what? Are the same. So for the cow, the alleles are similar. Now the question that we don't know whether they are both recessive or they are both dominant. So that's the question we have to ask ourselves. Let's quickly go to the next sentence. They say white is dominant over black, which means if they say the cow is white, now they say the white is what? Is dominant. It means both the 2 letters must be dominant. Both the 2 letters must be dominant because we say they are both the same. Now you go to the last sentence. They say use letter what? Q for what? For white. So it means for the cow we have capital letter Q, capital letter Q, to make it a homozygous dominant allele because both of them are the same. Now we go to the bull. The bull, they say, okay, fine, they say was crossed with a heterozygous white-faced bull. So even the bull is white, but now the alleles are different. We said once they say heterozygous, what does that mean? It means the alleles are what? Are different. So, but then they say it is white. White is shown by what? By Q. So it means for the bull, you will have capital letter Q. What will happen to the next capital letter? Will it be dominant or recessive? It must be recessive because the alleles are heterozygous, meaning they are different. So we have what? A recessive. Then this is what? This is the cow. This is what? This is the bull. So what does the question say now? Use what? A Punnett Square. You must also be able to underline this one. Use a Punnett Square. Sometimes they say use a genetic cross, genetic diagram. But in this case, they want you to use a Punnett Square. So now you take your alleles. Siyabuya starter. This letter Q, siufaka siyabuya starter. This one siufaka. So this part, it's for what? It's for the male. This part is for the female. This one is for the male. The signs that we sometimes use. But then let's look at it. How do we do it? Okay, fine. Now you see we have this square. But I guess siwathola njani lama alleles aphakathi nendawo, which are the offsprings, the F1 generation. Because let's told this alleles. Sicrosse the allele le, sayicrossa nale. Sabuya saba no capital letter Q, which is a homozygous dominant. Sisonke lapho. Kuze sibe nale offspring le. Sicrosse this allele, sayicrossa nalena allele. Sase siba nama alleles awu-2 kanje. So if you look at it thoroughly, you see that for this sihambisa ngalo hlobo. For this one sihambisa ngalo hlobo. Then we are done with this. But then let's check the next one. What about this one? Sicrosse this allele, sayicrossa nalena allele. Sathola in heterozygous. Sabuya sacrossa le, sayicrossa na yipi nale. Sathola another heterozygous. That's how we come up with this allele. So in a simpler way, we had Q like this way, like this way. Then we said we crossed this, we crossed this. So sihlanganisa le, sihlanganisa nale. So go onke uyihambisa ngale ndlela. It's simple. As we move along, we will see how we do it. But then let's check it. So with parents there, they're always there, but what we are looking at here, we're looking at the face of the color of the face of the animal. So with a white and a white, what about the alleles, the genotype, homozygous, dominant, heterozygous? Then we have the meiosis here. And meiosis, what is happening is the separation of the gametes, the separation of the allele, which results in what? In the gametes, which is represented by what? By the Mendel's first law, which is the law of segregation. Then here now we have the gametes. Then we do our cross, then we're done. That is the simplest part of it. Let's quickly look at the Mendel's first law. Mendel's first law, it's what is the law of segregation. They say this law states that the characteristics of an organism are controlled by pairs of alleles. These pairs of alleles, they separate randomly into different gametes during meiosis so that each gamete receives only one allele. Just understand it. It states that the characteristic of an organism is controlled by what? Pairs of allele. We have looked at pairs, We have looked at what? At pairs, we have looked at alleles. So it is controlled by pairs of alleles which separate randomly into different gametes during the meiosis so that each gamete receives only one allele. So it's what we have to understand with this one. But then we will change it. Mutation. Mutation. But let's understand it in in this manner. Just to give you an easy way of understanding it, segregation is linked with what? With separation. So we say they segregate or they separate randomly. Okay, fine. Let's have example number 3. Example 3. What are they saying in example 3? A farmer cross-pollinated a heterozygous pea plant with a yellow seed. Then basebenzisa capital letter G for yellow. Then they say we cross a yellow seed with a purebred pea plant with green seed. I want you to understand it thoroughly. What are they doing here? They are crossing a yellow seed that is heterozygous by crosser nani ne pure breed that is what that is green. Then sibabuya bathi uyellow sirepresenta ngani ngo G omkhulu. Okusho ukuthini sihlobo sami? Once they say yellow is shown using capital letter G, it means that yellow is dominant over green because we said a dominant allele is shown using what? A capital letter. So yellow, they use G. So it means G is dominant over what? Over green. I hope you get that one. Then we say the yellow one is heterozygous. So meaning, I'm sorry because you will not see thoroughly. So meaning they say the yellow is heterozygous. And they say use capital letter what? G. So it means you will have the capital letter G. You will have the capital letter G. What will be the letter next? Since it is heterozygous, will it be a capital letter G or small letter g? Think it's heterozygous, ain't it? So it must be a small letter g because they are not the same. You get it? They are not the same, they are different. So as a result, we have something like this. Okay, fine. Let's change. Let's look at this one. Now, they say you're crossing it what? With a purebred pea plant that is green. Purebred is linked with what? With homozygous. Masiti homozygous simply means that the alleles are what? Are similar or they are the same. So they say you use what? Small letter G. Since we say green is recessive, it means that you will have a small letter G, which is green, and another small letter G because they are purebreds. As a result, we have such gametes or such alleles. Okay, fine. Okay, fine. Okay, fine. Now let's have the first question. The first question says, indicate the genotype of each parent in the first crossing. Sesibheke le genotype ye parent yokuqala eyellow. Sayibheke genotype ye parent yesibili ethi ibe green. And then siyasho because you understand the genotype, check ukuthi is it homozygous or heterozygous. Once you understand ukuthi is it homozygous or heterozygous, bese ubheka ukuthi yipi I characteristic a dominant kulena enye. But sithi masibheka la sabheka ukuthi okay fine, for the two parents, parent 1 it's heterozygous, which is capital letter G and small letter g. Parent 2 it's purebred, which is homozygous, and it so our small letters anjani out 2. Then you get your 2 marks, free 2 marks. But go back, just reverse a little bit, check ukuthi how have you come up with the capital letter G and small letter g, which is heterozygous, and how have you come up with the small letter G, which is purebred homozygous recessive? Please. The next question. Use the Punnett Square method to determine the possible genotype of the offspring in the first crossing. So what are we are using here? It's a Punnett Square. So mabathi sayise Punnett Square sebenzisa yona singakutholi so wenze ngalo hlobo lolo. This type of crossing that we do. Use a Punnett Square. Okay, fine. Now, they say use a Punnett Square method to determine what? The genotype of the F1 of the offspring in the F1 generation or in the first crossing. Then remember I said you have the two, you put your gametes here. Sometimes file uhambisene ngalama steps lawana eswashoy, but here let's just do it like that. You have the gametes for this is your parent 1, this is your parent 2. Then we do the crossing. Sithola kanjani le? It will be this capital letter G and this small letter g. Lana ngicela uyiqaphele kanje. Ngaso sonke isikhathi sihlobo sami, i-dominant allele or i-capital letter e-representa i-dominant allele or i-dominant gene siyibhala ngakwa left. Ngiyangithola? Ngikuyakuthola wena kusasa so uyibhale kanje. This is wrong. So you always write it on the left. This is wrong. Let me erase it before you claim it. So people will say you cross these two, you get heterozygous. You come again here, you cross these two, you get another heterozygous. You cross these two, small letter plus a small letter, you get homozygous recessive. Small letter plus a small letter, you get what? Homozygous recessive. Homozygous recessive. So that is the simplest part of what? Of this crossing. Understand that equal uyihambisa kanjani. Les sihlangana nales. Siphinde sibuye futhi size kulesi. Sibuye les size kulesi okuqala. Les size kulesi. So that is the simplest part when it comes into this crossing. Do we have another question? I believe so. Calculate the percentage of underline. Calculate the percentage, Calculate the percentage of heterozygous offspring in the F1 generation. How many offsprings? These are your offsprings. Offspring number 1, offspring number 2, offspring number 3, offspring number 4. How many offspring anama alleles ati alabe heterozygous? Hetero means different. So uyabuya ibheka lot, okay fine. Le kule yokuqala awafani vele, so it's 1. Kule yesibili ayafana, ngiyayi-add noma ngiyi-add ngiyayiyeka. Kule yesithathu akafani nakhona, so it's 2. Kule yesi-4 asayafana manje, so ngiyayiyeka. So how many that are heterozygous? They are 2. Out of how many? Out of 4, because they say calculate. What are we calculating? Percentage. We multiply by what? 100. So it's 2 over 4 multiplied by what? By 100. Then you get 50. Are we done here? No, we're not done. It's 50 what? Percent. Then you get what? 2 marks. So that is the simplest part of it. Wena upewiti calculator any percentage of what? Heterozygous. If they say for homozygous, besizo yenza nakhona. But then in this case they say just calculate for heterozygous. Okay, fine. Then you have your answer 2 divided by 4 multiplied by 100, you get 50%, which is 2 marks. So in this case it's simple. You have 7 marks for free. That's why I say in this section you must get nothing less than 25 or 30 in question 4 of your paper 2.